Interpretation of lab results, Unit 3, Part 2, Immunohematology, also known as blood bank. Immunohematology is the study of the principles of immunology as they pertain to the blood cells in the plasma or whole blood in hematology. It is also known as blood bank. Why? Because that is a department that deals with the components, the packed cells, the plasma, the platelets, or the cryoprecipitate that are donated so that those products can be processed and transfused to patients. Excuse me. So blood products that are donated will be the ones that be processed and be ready, tested so that they can be used for human transfusion. Blood bank testing is very comprehensive, but we're only going to touch here on the very, very basic. We're only going to talk about the basic blood typing for ABO. ABO typing, meaning the antigens present from the ABO family that determine the blood type. The Rh, that you know it as being positive or negative, and that is another family of antigens, but we only concentrate on one, on the D antigen. But we will talk about what is involved in the cross-matching itself. But the proper medical term for that is compatibility testing. So compatibility testing will include finding out which ABO antigens the patient has so that we know that whether the patient is A, B, or O. The Rh type, or the presence or absence of the D antigen. If the patient has the D antigen, then they are Rh positive. If a patient does not have the Rh D antigen, then they are Rh negative. And we're going to be looking for unexpected antibodies to some of the most common antigens that can be naturally present on the membrane of the red blood cells, and the cross-match itself, meaning we're going to get a little bit of the donor's blood, so to mix it with a patient's serum or plasma to see whether there's a reaction. If there is a reaction, then that blood is not compatible. We should not give it to the patient because the patient might end up with a transfusion reaction. You have a little sketch there about the different components that are gathered from one unit of blood. If you have heard the commercials on TV, they say that you can save 3 lives with 1 unit of donated blood. And it is because every unit of whole blood is processed and separated into 4 different components. We have the red blood cells. Remember, we start with whole blood, and then the unit of blood will be centrifuged, and the packed cells, the red blood cells, are gonna go all the way to the bottom, like you would in a regular test tube, in a regular vacuum tube. When you centrifuge it, the red blood cells will go to the bottom, and they will be processed as just packed red blood cells. The plasma that is on top of the unit when the blood is centrifuged is going to be moved to another bag, and that will be labeled as fresh frozen plasma. And it is called fresh frozen plasma because it has to be frozen within 6 hours of collection. So it has to be processed and then frozen right away. And all the testing has to be done with it. And I'll mention more about the testing that is done on each unit. And then out of that fresh frozen plasma, they are also going to remove the platelets. And there's minimal amount of platelets, but they are enough so that if a patient, let's say a patient that is receiving chemotherapy, or they are taking some medicine that destroys their platelet, if they need those platelets to prevent bleeding, then they can just pull them in one bag, pull different bags, 5 or 6 bags, and give the patient a good amount of platelets to help them with bleeding problems. And then they can also just separate the cryoprecipitate. And cryoprecipitate is this plasma protein So it is a plasma protein, so it comes from plasma, and it is full of fibrinogen. So if a patient has problems with fibrinogen deficiency, remember that fibrinogen is factor 1, and that is the one that once it gets activated will become fibrin. So if the patient has a bleeding problem, if their coagulation has been compromised somehow and their fibrinogen level is too low, then they can get cryoprecipitate. Cryoprecipitate is also rich in factor 8. So hemophilic patients might also receive cryoprecipitate. So take a few minutes to read the information in this chart. Is pretty good information, and it should help you to make sense of what we're trying to do. Remember, there is more to this than what we're going to cover in 8 slides. So this is just so that you can get an idea of what the blood type is about. So if somebody tells you you're an A positive, what does that mean? What does being an A positive blood type mean? What does it tell you? So that's what you need to get out of this PowerPoint. So in slide number 3, it also gives you a little bit more information about what we're looking at. And remember, from the previous unit, the immunology unit, we know that we have self-antigens, and those self-antigens makes us unique, make us us, right? Just makes us, in my case, my antigens make Veronica a unique person. And those antigens are found in every cell in our body. The red blood cells are full of antigens. They have a bunch of them naturally, and they are based on genetics. So if your mom has them and your biological father has them, there are chances of you having them, having received them as either autosomal recessive or based on genetic inheritance patterns. Okay, so you get those antigens that make you you. And we need to find out what those antigens are when you need a blood product. Remember, the blood product can be packed red blood cells, plasma, platelets, or cryoprecipitate. The most important of these many different antigens that we have on the red blood cells, the ones that can lead to very fatal reactions are 2. The ABO family of antigens and the RH family of antigens. So whenever we need to give a blood to a patient, we want to know their blood type. And their blood type will include the ABO antigens and the presence of the D antigen that it comes from the RH family. This is key for you to understand. The antigens on the cells, the antigens on the red blood cells, the antigens on the platelets, the antigens on the white blood cells, the antigens on the cells give you your blood type. Antigens on the patient cells that come from the ABO family and the RH family will give you your blood type. As a precaution, Mother Nature, I don't know how this was decided, but whenever we're talking about the ABO family, if you have the antigen, if you have antigen A or antigen B, or both antigens A and B, or you don't have either A or B, you're missing both. A and B are not present, so you are O. Should be 0, but since the other ones are letters, they decided to say O. Okay, so we have all those 2 antigens of the ABO family: A antigen, B antigen. You can have both A and B antigen, or you can have neither. So those antigens will give you your blood type. If you are A, you will automatically, naturally make antibodies in your plasma against the antigens that are missing. So if you are A, that means that you don't have B. So your body will naturally make anti-B. And that means that we have to do compatibility testing or cross-matching to make sure that you are receiving that right blood product. Otherwise, antibodies in the donors units, and the one that was donated can attack your blood cells and can lead to a fatal transfusion reaction. So again, the antigens from the ABO family, the presence of the A antigen, the B antigen, or both the A and B antigen, or neither. If you don't have A nor B, the presence or absence of those antigens are going to give you your ABO type. The antibodies that are going to naturally be present in your blood, in your plasma, will be to the antigen, the ABO antigen that you're lacking. And that is what this slide says. Another part of the compatibility testing includes looking for other unexpected antibodies that may be found in the patient's serum or plasma that might be might attack or might be present and may cause a problem with some of the most common antigens that are found on red blood cells. The last slide, slide number 8, gives you a short list of the most commonly encountered, the most commonly found antigens on the red blood cell. And you see there's a lot of families. And there's more than that, because those the list on the slide number 8 gives you the families. So each one of those families might have 2 or 3 antibodies as part of the family. So just so that you know. So when we're doing compatibility testing, or the cross-match, that is a short name for compatibility testing. When we're doing the compatibility testing to make sure that the patient will receive an effective, a good product for them, includes the ABO type, the RH type, the looking for any unexpected antibodies that might be present in the patient's plasma or serum, so the antibody screen, and then the actual cross-match. Okay, I gave you some examples there of what it means, and hopefully that will clarify a little bit. Slide number 4, it talks about, again, it's very repetitive so that you can, the more you read it in slightly different ways, hopefully the more sense it is going to make. Compatibility testing, again, includes the ABO type. So we're looking for the ABO antigens, or the antigens on the ABO family. The RH type, and the RH type is just the presence or absence of the D antigen. If the D antigen is present, then you are RH positive. If the D antigen is not present, then you're RH negative. And then the actual, the looking for the antibody screens, antibodies against the most commonly found antigens on the red blood cells, and then the actual cross-match, which we get a little bit of the unit of blood, the donor's blood, and we mix it with a patient's serum to just make sure that there's no reaction. We don't want there to be a reaction, because remember that antibodies are their job of an antibody is to find its corresponding antigen and destroy it. And we don't want that, because it leads to a lot of problems. Another commonly done test in routine blood bank is the DAT, so the direct antiglobulin test. And that is looking for the red blood cells of a patient that have already, or that is suspected that are coated with an antibody already. Because if a red blood cell is already coated with antibodies, because of those antibodies have attached to an antigen on the red blood cell, remember that the antibody will try to destroy the red blood cell, and we do not want that. So if we suspect that there might already be antibodies attached to the red blood cells, we're going to do a DAT test just to make sure. Sometimes a DAT can be caused, a positive DAT can be caused by medication or drugs. drugs being legal or illegal. So legal drugs are medicine, illegal drugs are toxins. So it can give you a positive DAT. So we want to see whether there's any antibodies that are already coating the surface of the red blood cells that might lead to a hemolytic problem. And on the right, some drops of blood here. And this is just to illustrate, to tell you that if you have red blood cells, the donor is type O, has no A or B antigens of the ABO family, so they are type O. They can give red blood cells, right? This is only red blood cell, whole blood. An O donor can give blood to an O patient, an A patient, a B patient, and an AB patient. So that's why we say that O and O blood type is a universal donor. And we're talking about red blood cells only, okay? And an A patient, if the donor has the A antigens, that donor can give blood to to a patient that is also A, or to a patient that is AB. A B patient, or a patient that has a B antigens on the red blood cells, can give blood to a patient that is B or AB. And an AB patient, a patient that has A and B antigens on his or her red blood cells, can only give blood to another AB patient. Remember, we want to prevent hemolytic transfusion reactions because that can lead to a lot of problems, even death. So we have to be very careful. And this summarizes, slide number 5, summarizes the blood types. This summarizes how we determine the blood type of a patient based on the ABO only, okay? So if we are looking, remember, antigens on the red blood cells provide the information about the blood type. So you can either have A antigens, B antigens, both A and B antigens, or have neither of them. You're going to see it on the chart on the right, and you can see the different schematics. They have group A, they have those little sticky, like sucky cups, green, coating the red blood cell, right? And that is just to denote, just to give you an idea, this patient is A, has A antigens, so is group ABOA. The group B patient has this little yellow little the sticky thingies there. So that gives them that we know that the presence of those antigens, of B antigens, makes the patient be B type. If they have both, the green and the yellow, then they are ABO type AB. And if they don't have neither of them, nor A nor B, then they are O. So the presence or absence of the A and B antigen will determine your blood type. If you have A antigens on your red blood cell, you will naturally make B antibodies. That's why you have there the B antibodies right there. And it says antibodies in plasma, and then you have anti-B. If you're group A, you're going to have A antigens and anti-B antibodies. Okay? If you're group B, you're going to have B antigens on your red blood cells and anti-A antibodies in your serum or plasma. If you have both A and B antigens coating your red blood cells, you are not going to have neither A nor B antibodies. You don't want them there, because if you happen to have A or B antibodies, they will attach to the specific antigen and they will destroy the red blood cell, and we don't want that. If you do not have A nor B antigens, on your red blood cells, you will have both anti-B and anti-A in your plasma or serum. This chart, guys, This is a huge hint. If you understand this chart, you will have no problem with the homework for this unit. If you do not understand this chart, then you have to read the information over and over and over again until you do, and maybe make your own schematics so that you can understand where the antigens are, where the antibodies are, and what does it mean when you have one or another. I can tell you a test question could be, this patient has anti-A antibodies in their plasma. What is the antigen that they are expected to have on the red blood cells? So you should automatically know that if they have anti-A antibody, they will have the B antigens on the red blood cells. So the answer would be B antigens. So that is the application, but remember, it is expected that you will have the opposite antibody to the antigen that is present on the red blood cells. Okay, and if you don't have any antigens, you're going to have both antibodies in the serum. And if you have both antigens, then you're not going to have any of those antibodies in the serum. Then we move on to slide number 8. And this is the other part of your ABO and Rh type. I am O positive person. So I am an O positive. My blood type is O pos, right? So that means that I do not have A, I do not have B antigens on my red blood cells. I should have anti-A and anti-B antibodies in my plasma. The positive, that's for the O, right? O positive. The positive is determined by the presence or absence of the D antigen on the red blood cells. The Rh blood group is the second most important blood group when we're talking about immunohematology testing. It is very, very powerful, and if you get the wrong blood, it's not going to, it's really not going to be as fatal as having an incompatibility with the ABO, but it can lead to other problems, especially in women of childbearing ages, women that can become pregnant. Okay, so the Rh blood group is the second most important, and what it means when it says you're Rh positive, it just means that you have that D antigen on your red blood cells. And if it says Rh negative, if you're Rh negative, then you do not have that D antigen on your red blood cells. So an AB negative person, what does it mean? Well, it means that they have A antigen, B antigen on the red blood cells, and they are lacking the D antigens because they are AB negative. So they have A antigen, B antigen, but they do not have the D antigen that makes them Rh negative. Okay. The application of this and why we worry so much about this is because the patients can become sensitive, and like I said before, but it is in women, especially women that may become pregnant, that the Rh is very important. And slide 7 has a very cute, very nicely, presented, diagram of why we want to know about the Rh type of a young mother to be, because if the father is Rh positive, and this is biological father, okay? So the father is Rh positive. That means that his red blood cells have the D antigen, right? And if we have a mother to be and gets pregnant from this Rh positive guy, if she is Rh negative and they come together and they make a baby, there's 50-50 chances that the baby, the fetus, might be Rh positive, have the D antigen, or be Rh negative, or be not have the D antigen. If the fetus that comes out of this couple is Rh positive, meaning the baby has the red blood cells of this fetus has the antigen on the red blood cells. If there is any maternal fetal blood combination, if for some reason there's a little bit of mixture of blood between mother and fetus, then the mother can become sensitized. The mother immune system will wake up and say, these red blood cells have a D antigen that is not mine. These cells are foreign to me, are non-self. I'm going to have to make antibodies. And then the mother makes anti-D antibodies. And we have memory cells, right? Remember we talked about the immune system having those memory cells. Usually during the first pregnancy in an Rh negative mother is not a big deal. But if she happens to get pregnant again and that fetus is Rh positive or has a D antigen, then she already has the anti-D that she made from the previous pregnancy, and that anti-D, because it's IgG, can cross placenta, and those antibodies can attach to the fetal red blood cells and destroy them. And then the fetus will go through a hemolytic disease. It's called hemolytic disease of fetus and newborn because it can happen during gestation or after birth. So it can be very fatal to newborns and fetuses too. So that's why we worry so much. That's why this is the most important clinical application of knowing your blood type, specifically for a woman of childbearing ages. There is this immune globulin, this anti-D, synthetic anti-D, that can be given to Rh negative mothers. I'm sorry, I dropped my phone. Is given to Rh negative mothers during pregnancy and after birth to prevent them from making anti-D and to prevent from exposure from possible problems in other pregnancies. So this is, if you just look at the schematic and try to see the pluses and the negatives that are there, in the mother that are all over the place. The little Y's, the green Y's that you see and floating in the mom, those represent the antibodies. So this is pretty important, and we paid a lot of attention to that. Everything from determining how many doses of this synthetic immunoglobulin need to be given to the mother to just try to prevent this type of sensitization. It all falls in the laboratory. So it's a pretty big deal. And then the last slide I was telling you about, this is just to give you an idea of the different families of the different antigen systems that can be present on the red blood cells. That's why we do the antibody screen. We're screening for the presence of those antibodies in the patient's serum because the antigens might be present in the donor's cells. Remember, we do not want the antigen and the antibody come together because it can lead to a lot of problems. It can lead to a hemolytic crisis and complications with renal failure and complications with cardiovascular problems. So this is why we need compatibility testing. Remember, we're looking mainly we're looking for the ABO antigens present on the red blood cells. We're looking for the presence or absence of the D antigen that is part of the Rh system. And then we also do the antibody screen, that we're screening the patient's plasma or serum for any antibodies against the most commonly found antigens on red blood cells, and then the actual compatibility testing. I hope you enjoyed this. This is very short, to the point, and I hope you can figure out what your blood type is and know what it means. You want to know what those letters mean. If you are A negative, what does that mean? If you are O positive or B negative, what does those blood types mean if they ask you, so what does it mean? How do they know that I am O positive? Well, that means that you do not have the A antigen or the B antigen, but you have the D antigen on your red blood cells. That's what the blood typing is all about.